that's what we're. I will be chairing today's webinar. Now, for those of you who have never heard of the Human Remains Committee, we are one of several committees making up the Norwegian National Research Ethics Committees. We are ultimately under the Ministry of Education. And our responsibility is to outline ethical standards for the use of human remains in research and to provide independent ethical evaluations of proposed uses of human remains outside of the context of medical research. And this can be the research projects of the type we're going to discuss today. It could be the public display or exhibition of human remains. It could be repatriation of human remains, just to name a few. Now, it feels a bit cliched to open this webinar by highlighting the explosion of ancient DNA research over the last decade or so. But perhaps it just feels cliche because I find myself saying it so often. But to those of us who follow the field, it is readily apparent. It is equally cliche to say that it raises a host of ethical issues, but it does. Now, the most obvious of these is the pressure it puts on museum and university collections. But this explodes very, very quickly into issues of how one defines what question is worth spending such valuable material on, the comprehensiveness of data collection and how this will affect the need for future sampling, the storage of genetic data and how it will be done, by whom, and how to facilitate or limit access, how to protect the rights and address the concerns of affected parties, such as indigenous communities. Beyond that, we must think about the interpretive frameworks we apply to genetic data, when these are appropriate and perhaps inappropriate, how the narratives we build and the raw data itself can be used and abused when it enters the public sphere, particularly in a public whose view of the potential of aDNA is skewed by things like the genetic genealogy issue. Now, there are several key players in ancient DNA research. First, we have data producers. These are the paleogeneticists themselves. Then we have the data consumers, and these are the archaeologists, the anthropologists, who use the data to address their research topic. And certainly our experience in Norway is that the data producers are also data consumers. And what will typically happen here, at least, is that the ancient DNA laboratories or the paleogeneticists will have a specific question or set of questions they want to ask of the material, and they will link up with a researcher or in cultural heritage institution who has their questions that they want to ask of the material, and they collaborate forward on that. In addition to these groups, we have those who control access to the material, those with curatorial responsibility for the material, which might not be the same groups of people, and those with curatorial responsibility for the data. And the role of this last group represents, at least to my eyes, one of the biggest challenges for aDNA research. We'll get to that later. Right now, I'd like to introduce you to the panel of very accomplished academics who have generously agreed to help us look at these issues today. We have Professor Eske Willerslev. He's one of the most recognized names in the field of ancient DNA studies. He's an evolutionary geneticist and director of the Center of Excellence in GeoGenetics at the University of Copenhagen's Globe Institute. Birgitte Skar is an associate professor in the Institute for Archaeology and Cultural History at the Norwegian University of Science and Technology. She comes to this topic from various angles. As a researcher, she has planned several projects involving ancient DNA analyses. As a curator at her institution, she is involved in granting access to material, and she is also a former member of the Human Remains Committee and has vast experience evaluating the ethical component of research projects involving human remains. Martin Furholt is a professor of Protohistoric Archaeology and Social Archaeology at the University of Kiel's Institute of Pre- and Protohistoric Archaeology. And Martin is, to my reading, what I would call a critical user of ancient genetic data, which is to say he engages with the data, but has also written extensively on the limitations of the interpretive frameworks that typically get applied to such data. And finally, we have Kerstin Lidén. Kerstin is a professor at Stockholm University, and she's a bit of a jack of all trades when it comes to archaeological science. She was one of the early adopters of the aDNA methodology and helped introduce it to Swedish archaeology. So as I said, they're all extremely accomplished and bring a vast amount of experience and a broad range of experience to today's discussion. I think it's going to be a good one. Now, a brief description of how this is going to work. Each of our panelists will speak for about 15 minutes on their chosen aspect of aDNA research. After this, we will have a 30-minute panel discussion based on the themes they've presented, as well as some of my own topics of interest. And finally, we will end with a Q&A session where you can ask questions directly to the panel. If you have a question for the panel, you may submit it via the Zoom Q&A section, and I will pose it to the appropriate member of the panel. Please let me know if your question is directed at a specific member of the panel or if it's just a general question. So that's it. And now I'd like to begin with our first speaker, Eske Willerslev, who's going to talk about ancient DNA ethics. Eske, take it away.
Thank you very much, and thanks for the invitation. I thought maybe I could approach it by taking us through the process of doing ancient DNA, and then I can say something about what I consider of ethical concerns and importance at each step of the process, right? So if we start by we need to sample material, right? And typically it comes out of museums. To do ancient DNA research, it is what we would call destructive sampling in most cases. It means you have to take a piece of bone or teeth, for example, that will be lost forever in order to retrieve the DNA. And in my experience, I mean, there is already at that stage, actually, to some extent, this can be very often a disalignment, if you want, with in terms of interests. So as an ancient DNA researcher, obviously your interest is to retrieve the DNA and therefore, in principle, to get as much material or as well-preserved material as possible from a specimen, because that will increase your chances of retrieving the DNA. While as a curator, of course, you also have an interest in that, but you also have an interest in preserving things for the future, right? It's part of your job to preserve the specimen. So already there is, you can say, there needs to be some kind of negotiation happening. Additional to that, when it comes to sampling, there can be ethical concerns and considerations relative to third parties. I mean, I've worked a lot in the Americas. I worked in Australia, where there are indigenous groups who can have very strong opinions about whether ancient DNA research should happen at all. And there needs to be some kind of clarification. Who has the responsibility, right, to engage with the indigenous groups, right? And one thing is, it's important to remember that one thing is the law, another thing is ethics. Those two things do not necessarily, you can say, are not necessarily the same. For example, if you go to South America, in several countries in South America, and also in Mesoamerica, legally, it's actually only scientists that determine through a board whether you can get access to a specimen and conduct ancient DNA research. But the wider community, many in the wider community would argue, well, you need to have tribal engagement, right? But it's not a legal demand. But when you are facing, let's say, publication, trying to publish afterwards, you might very well be in a situation where they ask, well, where's your ethical consent from the appropriate indigenous peoples? And you might actually face problems of publishing unless you have that in place. So then, and I've seen also in the States, for example, that maybe curators have given away specimens to DNA researchers, but without taking into account that they need to engage with the appropriate indigenous peoples. So there needs to be a clarification in terms of what needs to be done on that regard, who will take the responsibility to do it, and what kind of, you can say, approval, if you want, is needed from the indigenous peoples. And it sounds pretty simple, I've experienced several cases where that is not actually, you can say, taken care of properly. Okay. Then after that, of course, you have a DNA extraction, and you have the generation of the DNA sequencing material. And there, I think, a lot of non-DNA people, if you want, are not necessarily aware that there's different types of production of data. And these different types of productions will put limitations to what you can do with the data. So in other words, you're making a destruction of a sample, right? But it varies what type of information you can actually retrieve from that specimen and from that destruction, depending on what kind of methodology is applied, right? So some people, would go for what is known as mitochondrial DNA, for example, right? And mitochondrial DNA, it's quite limited, you can say, in terms of the information you can get out of it. It says something about, you can say, the mother line, if you want, right? But you can't get anything about, let's say, any traits out, for example, right, on the person, what kind of eye color, hair color, skin color, whatever, risk of disease or whatever it could be, right? It's also very limited in terms of in terms of what kind of demographic analysis you can do, for example, right, with mitochondria. So if you make a destruction simply to do mitochondrial sequencing, you have to be aware as a curator that you have made this destruction without really getting the full potential of the sample in terms of DNA recovery. The same, then if you go to genomes, which is kind of what has been produced over several years, there's genome-wide capture, right, where you are capturing, you can say you're trying to capture, certain substitutions or certain places in the DNA that can be informative of different things, but you're not sequencing, you're not attempting sequencing the whole genome. Okay, that can give you information. It gives you much better demographic information, but and it can also give you information on phenotypes if you have captured SNPs that are informative in that regard, right? But you have to remember that type of data will not give you two quite important things. First of all, if you have, there's new SNPs being recovered all the time, who can say something about phenotypes and disease risk and so forth. And of course, if they are not part of the capture, well, you don't have that information, right? Secondly, you are not getting what you can get with full genome shotgun sequencing. You're not getting things like pathogens out, right? The infectious diseases that these people had. So the last kind of level is what we will call whole genome shotgun sequencing, right? That's where you're trying to achieve or sequence the whole genome, as much of it as you can by shotgun sequencing. And that, in principle, is the approach that can give you the maximize the amount of information you can get out. You will get, whatever is possible in terms of SNPs also for the future, whatever will be discovered in the future. You will get, you will also capture at the same time, you will get sequencing the pathogens that might infect, like smallpox, plague, whatever, Borrelia, whatever these people have had in terms of bacterial infection and DNA viruses, right? But even there is different levels, right? Because then you talk about coverage. How many times have you sequenced the genome? How much coverage is there around the genome? And how much coverage you can get is dependent on the preservation of the sample, to some extent, the complexity of the DNA, but it's also dependent about how much money are you willing to put into the effort. And the higher the coverage, if you want, if you can get very high coverage genomes, if there's good enough DNA preservation. That will allow you to make very sophisticated models, for example, of divergent times. When did different groups diverge from each other? What was the amount of gene flow and the timing of the gene flow between different groups? And if you get sufficient coverage, you can even go in and look at epigenetics. I mean, which is, how is the genome mutilated, which again can say new things about traits. It can say new things about disease risk and so forth. So in other words, what I'm trying to say here is that DNA sequencing is just not DNA sequencing. There's many different levels. And I think when you make your assessment in your committees around the world, where you decide, well, should we go along with this group and their request, etc., right? You need to take that into account. So you need some people who has a basic understanding, so well, what can I expect and not expect, right? The other thing that has happened a lot, at least in the past, I would say, is that the ancient DNA researchers have come and said, well, we would just like more or less all your skeletons, right? There's no hypothesis, there's no timeline in terms of when should they produce results. And then another research group, ancient DNA research group, is then approaching, half a year later and saying, oh, we would also like some samples. And the answer is no, you can't get them because we have already given some to another research group, right? But what are the questions they want to address, right? What are the questions? The questions could be very different from the two different groups, right? And I experience, you're arriving and saying, well, please, would you like to collaborate on this? And the answer is no, we gave it away to this other group. And then you're waiting maybe almost 10 years before there is any results coming out, right? So I think it's super important that it's very clarified, from when you can say, consider these requests. What type of sequencing do they plan to do? Are they maximizing the information? And if not, why, right? And what is the timeline for publication, right? You could come and say, well, if you haven't produced anything after X amount of years, you have to deliver the sample back, right? And we can give it to somebody else, for example, if it's precious samples, right? So I think these things needs to be in place. Then, of course, there's then the analysis part afterwards, right? And there, I think you also need to make up your minds. I mean, how much do you want to be involved in this part? Right? Because it's of course totally understandable that you are not able necessarily to undertake the analysis. It's very sophisticated and very specialized. But that doesn't mean you can't be participating in asking the right questions, right? And also putting the right context to these questions. And there you also see a variety of approaches in the ancient DNA community, right? There's people who basically just say, well, we just want, our genetic data, and then we interpretate on that. And that's it, right? And then you have groups that are much more multidisciplinary in their approach and actually bringing in, you can say, expertise within archaeology, anthropology, etc. So you also need to make up your mind. What do you expect in terms of participation, right? And you need to make up your mind in terms of, well, what is actually demanded in order to be a co-author on the publication. There you see all kinds of, again, different approaches. Some people are kind of saying, well, we want to be on, and the only thing in principle they're doing is they're opening the drawer, right? But to get your sample, we want to be on your paper, right? And that's actually ethical. I would say it's poor scientific practice, to be honest, right? Then you can say, well, if I'm giving the sample, I want to be involved. I want to be involved in the scientific discussion. I want to provide background, etc. And that's a different matter, right? Then you're actually participating really in the study. But I think these things needs to be clarified as well, right? Then finally, then you're reaching a step on the publication. And there, I've also experienced and heard about many different approaches, right? I mean, there's some that get a paper and then they have 3 days to say, well, do you want to be on it? Can I improve it or not? Right? And then it's kind of go. This is not obviously the way, right? I mean, you could demand also, if you want to be involved, that you say, well, I actually really want to be involved. I want to have critical input to the paper. I want some time to read the paper properly, discuss it, etc. Right? To make sure that you are being properly enrolled in the study. Then finally, in terms of storage of the data, I mean, the tradition, I would say, within ancient DNA research is that everything becomes publicly available. And it's a principle which, as natural scientists, we really like, because it means that everybody can get access to the data. Everybody has the possibility to check for reproducibility of results, right? It can be, you can say, the data can you can say the utilization of the data, if you want, is maximized, right? Other people might have great ideas or new ideas, and it can be used in new ways. So this has been really, you can say, the golden standard in ancient DNA. But this is to some extent being challenged now, particularly with indigenous groups, right? Because you go out and make an agreement with indigenous communities of how this data can be used. You say, for example, well, the purpose of this study is demography, right? This is what we want to do. And then somebody else download it and use it for medical studies, right? Or as I've experienced more recently with our Native American studies, then it's downloaded by these ancestry companies, right? That are making tons of money in terms of saying, well, are you related to the Kennebec man or not, right? And the tribes are getting hundreds of requests every week from people who say, well, I should be enrolled in the tribe, right? Because I actually are related to somebody, right? And the other thing is that the mythology in ancient DNA is developing. So when we started this with the type of genomes we were producing from some of these groups, you couldn't address things like inbreeding or effective population size. You can do that now, even from very low coverage data. And suddenly, you are facing a situation where you could say, well, that could be really within the frame of demographic studies, right? But it might be something that the stakeholders, such as the indigenous peoples, don't want. Because they are not interested in people saying, well, you came from this inbred population or whatever it is, right? If that's what it shows. So I think so now, for example, we have basically had to approach the communities again in America and said, well, there's some new databases being generated that are run by indigenous researchers, where you can say that to get access, you will have to apply. And then these you can say gatekeepers are then approaching the communities and asking, are you okay with this and that, right? And this is because they are super annoyed about particularly these you can say ancestry companies going out and using the data and making money from it. So these are some of, I would say, the ethical concerns that I have faced and experienced during my time as a researcher with ancient DNA. Thank you.
Thank you very much, Eske. That was fascinating. And I know we're going to have to come back to you with during the Q&A and during the panel discussion with more technical questions.
Sure.
Now I'd like to move over to Birgitte Skar, and she's going to talk to us about aDNA research on human remains, practical, ethical, and collaboration implications, a perspective from a Norwegian university museum. Birgitte, the floor is yours.
Can you hear me now? Yes, good. Yes, today I've chosen to put on the hat of the University Museum because I thought that was what was asked for, really. So let's see, I have to put on this. You can see the presentation. It says my internet connection is unstable. Can you see it?
Yes,
Good.
we can see it.
Yeah, good. I think before I start talking about today's issue, I would like to present just a few examples of what has been achieved doing DNA analysis on Norwegian material during the past 10 years. It's important to mention a few examples of that, I think, because in general, the museums are very pleased about the fact that our collections are being activated. If we start on the left corner, you will see that there's been some analysis of the some of the earliest Holocene material that we found in Sweden and Norway, and we now have a much broader understanding of the first peopling of Fennoscandia and the likely migration routes. And these studies are combining aDNA research, technological lithic analysis, stable isotope analysis, and radiocarbon dating, and they were led by Torsten Günther from the Department of Organismal Biology in Uppsala. Later, these studies have generated a lot of archaeological studies, and the latest being published last year by Mannen et al. Then we have, if you go on to the right to the two portraits, Younger Iron Age and the Viking period has been studied through several projects, notably through Magarian's work at the Center for GeoGenetics, I think was the name at that time, illuminating the biological impact of Viking mobility in regions subject to Viking raids and trade, as well as secondary effects on population admixture back in Scandinavia. These studies have both before and after been supplemented by stable isotope studies and have contributed to discern life history and social implications for the Viking society. And then down in the right corner, you can see an example from a publication. A number of studies based on human remains from the medieval cities, and particularly Trondheim, have kept coming based on Gilbert's sampling. Tom Gilbert is from the Globe Institute in Copenhagen University. And these analyses were already initiated in 2011, but have been supplemented with new samples from the MedHeal project that's run from Trondheim. And what we get is a broad spectrum of aDNA approaches, including epigenetics, actually, and pathogen analysis that are given a detailed understanding of demographic development and effects of the second plague pandemic and other diseases. Variable climate data, stable isotope data, and archaeological analysis, we will get, they're not quite finished yet, but we will get a much deeper insight into consequences of medieval city life on health and social change. And then we have some new project initiatives, Eske, that is from you and your institution, that will throw light on important processes of social change and health on the period from the late Neolithic into the Younger Iron Age. And these are forthcoming projects. I mean, all these projects are based on sampling of human remains that have actually been resting and sleeping, basically, in Norwegian collections for up to 100 years. So I think these projects are, to a very large degree, fertilizing new research. And now I'll get on to the university museums and our responsibilities. The 5 university museums in Norway their functions are anchored in several national and international acts and conventions, as you can see in the first point. The universities of Oslo, Bergen, Stavanger, Trondheim, and Tromsø have a particular responsibility to maintain and manage scientific collections and exhibitions. In effect, the 5 university museums in Norway function in collaboration as a national museum, but responsibilities are decentralized. The university's responsibilities entail facilitating and performing research on very large collections. I do not know the number of items in all the museums, but I know that our collection in Trondheim includes approximately 1.8 million artifacts and approximately 4,000 human remains, individuals. The university museums have a particular responsibility for archaeological remains from prehistory and the medieval period, but also Sami archaeological remains, numismatic materials older than 1650, as well as maritime archaeological objects. Material from the museum's cultural historical collections is a non-renewable scientific resource material. We do, however, afford possibilities to sample, as you now know, and analyze material from researchers from research institutions. It's not just everybody that can apply to sample our material, but you have to be affiliated with a research institution. And these institutions are national or international. We're receiving quite a number of international requests. And providing a good project plan and that permission from the museum's analysis review committee can be granted, you can get to analyze material. The committee at the museum seeks to balance knowledge needs against preservation issues, and of course is also very concerned with the general research ethical principles. As we saw that the number of applications raised some years ago, the Norwegian University Museums developed a set of guidelines for application and processing of requests for analysis. So we have a common set that functions with all the university museums in Norway. Sampling and analysis of human remains have a particular focus in these guidelines. And that involves approval by the National Committee for Research Ethics on Human Remains. And this happens prior to the university museums making decisions. In the very few cases where we have requests for sampling on Sami material, the Norwegian indigenous group, the Sami parliament is involved in decision making. I should also say that you can check on these links and also find the National Ethical Committee's set of guidelines. I do not find time just now to go into these in detail, Sean, but maybe you can fill in later. And there's I think 9 points of ethical aspects that should be evaluated for each project request. So what I've done, this is a very simple little calculation, but what I've done in order to get an impression of the character and amount of aDNA research on archaeological human remains in Norway, we've used the public archive of the National Committee for Research on Human Remains to access the committee's responses to applicants, not the applications, because they may be kept secret or private, but the responses are public. And these letters are very detailed regarding the contents of the requests. First, we were interested in getting an idea of which collections receives most applications. From looking at the pie chart, there's no doubt that the University Museum's collections are the ones receiving most applications, followed by the Schreiner collection, which is the dark blue here, at the University of Oslo. And since the Schreiner collection also holds material from the University Museum of Oslo, we have extracted the applications that are concerning prehistoric and medieval individuals and calculated them with the university museums in this diagram. You can see that actually the only other institution that receives requests is the Svalbard Museum, that has a small contribution here with 6 individuals. Now, from this statistics, we do not know. We know whether the ethical committee said, yes, we think this is a good idea, or no, we think you should consider revising your application, or no, we do not think you should do this study. That we know, but we do not know whether all these requests actually led to research. So we would have to go deeper into the material to find out that. Sean was mentioning that DNA research is accelerating in the world, and the number of samples and results are phenomenal. I think in a meeting just a week or 2 ago, I heard the number 50,000 sampled genomes in the world, but that was just sort of an offhand number. I think when it comes to Norway, we have to look at these numbers. These are the requests coming into the National Ethical Committee, and in fact, the numbers do not indicate that there's such a rise in requests. I think what we can see is that aDNA projects come in waves depending on national and international funding, and depending on the relevance of including Norwegian material. There may be a large project just around the corner to the right here, but we don't know of that project as of yet. So the numbers are actually not indicating a phenomenal rise. The top in 2017 is caused by one large international application for research on more recent material held at the Schreiner collections, and whether this sampling actually took place, I do not know. They will know at the Schreiner collections, and I do not know and have not been able to find the results published anywhere. This was a material on a reasonable recent set of populations. Without this particular application for 300 individuals, the general trend regarding sampling of Norwegian archaeological material for aDNA analysis is actually stable or possibly falling. And then again, we have to remember that it's not all these requests that were actually reproved, but this shows the number of applicants. Then just to talk briefly on the museum's concerns regarding collections and data. This has already been mentioned partially by Sean. But I can tell that when I took part in the sampling for the Viking Genomics project at the Schreiner collections in 2016, the head of collections, Professor Furholt, said and sighed, "This will be the last time in the next 50 years this collection is sampled." Because by then there had been several projects, particularly doing isotopic studies, but also, for example, earlier DNA studies. I'll say we've seen the same tendency in Trondheim. Some collections are very popular, some are much less popular, or people don't know about them. And in Trondheim, we see that certain medieval grave sites are very important. The projects want individuals from these grave sites, and very often the same individuals. And in Trondheim, this is mainly due to the material being reasonably well preserved, but also because the stratigraphic stratigraphical dating is good. And in this way, the projects can avoid actually spending money on radiocarbon dating. I think if we proceed in this way and permitting these projects, then we do risk to exhaust the most well-preserved material. And we do know that we do have relevant material for the application if the person is willing to spend money on dating, for example. So the university museum's prime concerns are with preservation and legal matters of ownership and ethics. The museums find it challenging to find good solutions to maintain their role as long-term managers of data that, according to our mandate, belong to the Norwegian state. Presently, generated data from aDNA projects are stored with the European Data Repository, granting open access, as Eske was mentioning. The ENA, for example, is one such repository. And as I've understood it, very often the raw data are not stored with these repositories, but are stored with the analyzing lab. In the most recent MOU, Memorandum of Understanding between the University Museums and Lundbeck Foundation GeoGenetics Center, these issues are addressed very clearly, both regarding generated data and raw data. And I think we found very good and satisfactory solutions to this. This means that also the museums can request access to the raw data, contacting the analyzing lab. The MOU also precludes commercial sale or redistribution of data to third parties. But still, some university museums are very restrictive regarding involvement with DNA initiatives. And that is because they wish to keep track of their data, a complete track of their data, and they wish to have the possibility to perform a renewed ethical evaluation when a set of data is reused. One is concerned that this may, as Eske was mentioning, downloaded by some medical company or somebody having another type of interest in the data, and that data might be abused. So this is difficult to preclude in an open access world. Finally, I'll talk a little bit about collaboration. Eske was also mentioning this. The university museums are very aware of the huge impact of aDNA analysis on human skeletal collections for actually fertilizing prehistoric and early historic research on prehistoric and early historic societies. These projects in many cases activate collections that may not have been analyzed for several generations, and in a very different way. At the same time, it becomes very imperative to educate our own research staff to understand more of the methodology and be able to employ results from this type of research. We are, after all, also research institutions. One approach used by our museum and several of the other university museums in collaboration has been to enter into a more direct collaboration when approached by international labs that are asking for permission to sample. This approach not only pertains to aDNA projects, but to a wide scope of projects using cutting-edge natural scientific methods in analysis of archaeological materials from our collections. A challenge is, of course, as you may feel, Eske, that the international projects are often big data ventures with a set agenda, a set consortium, and a financial frame. While our research interests are geared perhaps more towards in-depth archaeological and anthropological studies of societal processes or particular sites in a more confined geography. But these collaborations are also covered by a memorandum of understanding, and we're only just starting to gain experience on how it's going to work. We will see in your new project initiatives, Eske, how we're going to be able to collaborate. And of course, these national projects also need funding. So I mean, the scope of the national projects are very dependent on and the way we can contribute to the big data projects is very dependent on us getting funding for that sort of thing. That's what I wanted to say. Thank you very much.
Right, Birgitte, thank you very much. That was a very very good overview of the situation in Norway, and I was glad to hear that we're not being quite depleted of all of our samples quite yet. Got a wave of requests for material. Excellent. Now we're going to move over to sort of the other end of the spectrum, move away from the technical aspects of this, and we'll move into the interpretive aspects of this. And we're going to have Martin Furholt. And he's going to discuss ethical dimensions of the use of aDNA data for archaeological interpretation and narrative building. Martin?
Yeah, thank you very much. Oh, one moment. So, do you see the full screen? Okay. Yeah, so what I'm going to talk about is something that I have been discussing a lot here in a Center for Advanced Study during the last year, which I was heading together with Daniela Hoffmann from Bergen University, which was actually dealing with especially migration narratives in the context of use of aDNA data, but also beyond that. So as you, Sean, introduced me in the beginning as a critical user of aDNA, I would also like to state that I'm actually also a quite enthusiastic user of aDNA studies. And where you will see I will be using the third millennium, the step migration discussion, third millennium in Europe, step migration example as maybe one example illustrating many of my points. And this is actually also a field that I've been working in for many years before DNA actually became important, and the impact that DNA has had on this field is very exciting. I mean, we are really now, we have the possibilities to talk about mobility and about migration, something that was neglected for a long time for many reasons. So these are new topics that are only now possible really to talk about. In recent years, we have been turning also more to social organization, kinship structures, also aided by this very fantastic new data set. There is also discussions about gender and family structure and all these things that I will talk about in a moment. But of course, while we do this, while we make use of DNA data and construct our archaeological narratives and engage in archaeological interpretation, or I would call it archaeogenetic interpretation, then of course there are ethical issues that we have to take into account. And I think the third millennium, this step migration example, is something which can show, as the discussion has gone the last 5 to 7 years, it is probably showing very well how narrative constructions can have political implications, especially when it comes to the way that it is presented to the public in public dissemination. And I think this third millennium example shows that what was discussed, part of what has been discussed, especially in the early papers when just this kind of new data set became available and migration was the big topic that came out, was that certain or rather specific views about identities, ethnic identities, ethnicity, about migration, but also about social organization, gender roles, and kinship, as I just mentioned, that they have been put forward in a way that is in a way problematic, at least should be discussed as such. To start with the first one, I mean, this was really the big reveal when it came to third millennium Europe, that migration actually was an important factor shaping the archaeological record and thus also the prehistory of of Europe. But the way in which migration was used, especially in the beginning, was a rather stereotypic concept. It was this kind of mass migration narrative where, and it coincided in 2015, of course, also with the refugee crisis in Europe. So it had a lot of kind of feedbacks to the modern experience. And it was somehow portrayed in a way that easily somehow fed into certain political currents. So the idea to see migration first and foremost as a kind of threatening phenomenon where, the local identities are being overrun, this as a kind of violent and disruptive process. This is something that, was then also taken up in these discussions, in the political and social discussions back in the days, 2015, 16, 17, and so on. And it is problematic because migration was, despite it being talked about as mass migration, it was basically left unexplained. It was not explicated. It was not, tapping the kind of anthropological, historical sources of knowledge that we actually have about migrations. And this also has to be seen in context of the ideas about identities and especially ethnic identities that were transported. And I think what worried archaeologists most was the use of the biological concept of population and the way it was somehow connected to the archaeological concept of archaeological cultures. And this is something that we as archaeologists have been struggling with a long time, because we have this unfortunate tradition that goes back to the early 20th century, when people like Gustav Kosinna was basically creating these units, archaeological cultures, which basically meant prehistoric peoples. So specific human groups, culturally and biologically homogeneous, were basically the main players in prehistoric Europe, or seen as being the main players in prehistoric Europe. Clearly, of course, modeled after this kind of mainstream thinking about nationalities or national ethnicities during the 19th century. And what it shows is, of course, a conflation of this archaeological term, archaeological cultures, and social groups, or a conflation of social identities with genetic ancestry. And this is something that we saw coming back in this discussion. I mean, if we go back to the archaeological and anthropological discussion about identities and ethnicity, it took us very long to somehow overcome this very initial idea about ethnic groups being, racially and socially homogeneous. And it was deconstructed, for example, by Franz Boas in the early 20th century, who actually himself was measuring skulls and all these things until he empirically could show that basically there is no ground for any biological connotation or biological basis for ethnic identities. And I think anthropology in general, Frederick Barth, for example, have clearly shown that ethnic identities has something to do with subjective self-descriptions, that they are subject to historical change, of course. And also for archaeology, very importantly, that ethnic groups are not necessarily marked by a common set of material objects. And if they are marked by material objects, it's quite selective. So it's very hard in hindsight for archaeologists to actually, pick what kind of material objects would be associated with ethnic identities. And of course, also it is clear that ethnic groups are not necessarily territorial, they're not static, they're actually quite fluid. And thus, archaeologists, if we now go back to the third millennium in Europe, we use archaeological cultures as a term that is denoting specific sets of material culture, sometimes also burial customs. In the case of Yamnaya, it's mostly burial customs. In the case of the Cordetware culture, it's also burial customs, but also sets of material culture. But it's still always clear that we are talking here about a kind of subset, a kind of sample that is not a kind of representative sample of a population in a specific region, but it is the archaeologically available sample. And it's also in this case, I guess, when it comes to these single burials in deep burial pits or covered by burial mounds, it's also a sample that is very, very favorable for aDNA sampling, because the preservation of human bones is better than in other cases. So it's basically a skewed picture about a very selective picture about populations in the steppes and populations in Central and Northern Europe. So it was then a little disturbing when we saw in these early papers that basically archaeological cultures were somehow the main building blocks to compare populations with, as you can see in this mixture analysis where samples are grouped after archaeological cultures, and these then compared with each other in terms to determine what kind of mixture processes would have taken place. And then, of course, by many different authors, we have this kind of what I call the dominant narrative, where this also was then connected with a kind of idea or narrative about this third millennium steppe migration being associated with a kind of aggressive intrusion of a group of male warriors from the steppes, and they were portrayed as warlike, but also patriarchic, top-down chieftain systems who would come into Central and the Northern Europe and marry the local women, somehow get rid of the local men. It's very violent, gruesome picture that is painted. Of course, a narrative, that's what narratives do. They work with debatable premises, of course, and which is a good thing, and that's why we are talking about this. They work with data that can be differently interpreted. And of course, the narrative technique involves also that you fill a lot of gaps which you don't actually have data for, but you somehow have to create such a kind of coherent story. You have to somehow also fill a lot of gaps. And this can make it highly speculative. For example, if we think about the social organization that I was mentioning here, this kind of top-down chieftains leading these kind of warrior bands of young men, this is something that's not really deduced from the archaeological material. Although, I mean, it is true that male burials dominate in this Yamnaya and early Courtois data set, but still the very explicit social organization here actually comes from a kind of reading of much later Iron Age written sources, which refer to this kind of Indo-European myths. And as you all know, Yamnaya and Courtois is often associated with Indo- or Proto-European languages. And so there is this kind of idea that there is some kind of pan-Indo-European social structure which stretches more than 2,000 years, which of course is a little problematic, especially if you look into the archaeological evidence, where, of course, with the Courtois, for example, you have a new focus on battle axes, so a specialized weapon, so this kind of warlike or whatever more bellicose motive is there. But if you look at the social structure, you have this, like here in Lundbeck, just as one example, you have this kind of large fields of small burial mounds with very moderate burials where people have maybe one battle axe and a set of personal items. So it's not really any social differentiation visible. If battle axes would be seen as status symbols, which they might have been at different times and different periods, this is rather a situation where everybody has access to such a status symbol, really then somehow deconstructing the idea of such things. So rather an egalitarian society where there is maybe even more autonomy of individual identities than there is this kind of top-down leadership that has been implied. When it comes to Yamnaya, it's even more striking that the idea of this kind of warlike Yamnaya steppe herders really is not reflected so much in the archaeological record. We do not have many weapons in these graves. Most of them have no finds whatsoever. And the idea that there is a higher level of violence associated with these burials is, more or less asserted rather than there is data driven. So I think what is the main problem for me here is not so much how this one specific narrative has been constructed, but rather what it implies basically about human societies more in general, because it does imply and has been interpreted by people in a way that this is basically, so if we have this kind of situation already in the 3rd millennium BC, then it must refer to a kind of more universal way in which history has unfolded. And this universal way would then look like this, basically that you have societies who are more or less organized in a hierarchically top-down manner, and that we have these groups, these cultural groups that are basically biologically distinct from each other, that we would have a situation where it's basically violent men who are active agents of change, and everybody else, women, but also peasants, for example, are always discarded as, you know, unimportant and really have no agency of their own. And that these groups basically are fighting each other to extinction in a kind of perpetual clash of civilization, and the most violent and ruthless group basically prevails. And I think, unfortunately, this is what has been taken up by all kinds of right-wing groups. You can see this on the web and in commentary places for more serious sites also, that people are saying this is hard science proof that, you know, all these liberal or left-wing ideas about individual creativity or the possibility to change identities or have multiple identities, that this is all, you know, now disproven by aDNA studies. Although it is clear that it is not the aDNA data that is implying this kind of narratives, but it is, of course, the narrative construction by archaeologists. And the use of concepts that come back from the early 20th century, which actually have in themselves the seed of this kind of idea of clearly separated populations and cultural groups. Just very briefly, to go away from this kind of ethnic identities, I mean, this is about the biologization of identities, of social identities, which is always a kind of threat when you are looking at those phenomena through the angle of biological ancient DNA data. So there is always this kind of danger. And a good example of this is also when it comes to the idea about family organization, kinship organization. This is the case from Eulau in central Germany, published already in 2008. This is a case where we have several cohorts where individuals who have been killed and they have been laid down together in groups of 3, 4, or 5. And what was published by Haag and colleagues was especially this grave number 99 here, where we have a possible nuclear family in the sense that haplotypes between grown-up male and grown-up female and children basically would add up to be a earliest nuclear family. And this is a very good article, and they are very nuanced in their discussion about alternative configurations who are also present. So there's, for example, this other grave here, 898, where we do have another kind of nuclear family-like configuration of individuals, but the adult female here is definitely not the biological mother of the 2 children. So it shows that basically, if we are talking about identities and the relation of, you know, family, the concept of kinship and family towards biological relatedness, this is a fantastic example about how varied those concepts might have been in the 3rd millennium. But what was taken out of this, and this is not the fault of Haag and colleagues, but I think somehow the way we are reading very selectively our data is that this site shows that the nuclear family as we know it today has been the standard more or less since the 3rd millennium BC. And I think this is just a good illustration that shows us what it is really maybe about. It's not so much about if I like one archaeological narrative or I dislike the other, but it is basically how we are very eager to somehow accept and not so much critically engage with certain premises and certain ideas about how the past could have been, usually the ones who portray the past in a way that we expect it to be, rather than, you know, having the possibilities to take this data and try to think in new ways and unexpected ways, rather than just, you know, confirming what we think about human nature, for example, or, you know, the aggressiveness of males and all these things. I think this is what it should really be about. And to end now, this was some critical words, but if we look at now the last 7 years, I would definitely say that this kind of discussions between archaeologists and geneticists, and also the, you know, much more fine-grained data that we are now currently getting, also has definitely shown a lot of or brought about a lot of progress in these kind of discussions, which, for example, the work here of the Stockholm group in southern Germany shows, where more small-scale developments over the generations have been shown in the third millennium, or the work here of the Haag group in Bohemia, where they could show a kind of diachronic, very complex picture of human movements and more complicated developments between different populations who are also transcending these kind of culture labels that initially were put in such a central role. And I think this shows that both a critical reflection of the concepts, but also, of course, more fine-grained data and advanced analytical tools can work together really to bring this field forward. Thank you very much.
Wonderful. Thank you very much, Martin. That was very, very interesting. Now we're going to move on to our final presenter, and that is Kerstin Lidén, who will talk about neither fish nor fowl, aDNA on the borderland between science and the humanities. Take it away, Kerstin.
Okay. So now you see my screen, right? Okay, thank you very much. So I'm the final speaker of this discussion seminar. And as Sean said in the beginning, I'm sort of a jack of all trades. And we've all sort of interpreted our own role in this seminar, so and slightly differently. And we don't know exactly what the previous speaker would talk about. So it might be some repetition, it might be something that we don't agree in, and maybe I'm a bit provocative, but I think that's also something that someone should be in this kind of seminar. So the title is Neither Fish Nor Fowl, aDNA in the Borderland Between Science and the Humanities. And I'm a professor in archaeological science, and I come from this particular borderland, both in terms of having worked with aDNA for a very long time, but also worked with other techniques. So I would just like to start to say something about, because there might be people out there who are not archaeologists at all, to say something about what is archaeology. Well, from my perception, it's an interdisciplinary discipline that studies humans and human activities during millennia. Archaeology is both a practical and a theoretical discipline, since it combines empiricism, theory, practice, and addresses contemporary questions from a long-time perspective. And natural scientific methods are an integrated part of the archaeologist's toolbox. And I would like to emphasize this. So what is aDNA? Well, most of you know what DNA is, but it's actually part of the humans that we do study, as well as being part of their activities in terms of providing traces of their prehistoric activities. Eske was talking of paleopathology, of microbes, for instance, and also Birgitte was talking about that. So it actually is a natural part of both archaeology and human studies of humans. So, and then I would like to say a little bit of, since we are now on Zoom, we're sitting in different environments, and I'm sitting right here in the Wallenberg Laboratoriet at Stockholm University. This is the Stockholm University campus, and this is the Vetenskapsakademin. It's the Royal Swedish Academy of Sciences, and it looks like this when you see the building. And why do I say anything about that? It's because that I want to say something about the interdisciplinary character of archaeology, and it goes very, very long back, and it goes as far back actually as when Willard Frank Lippig became a Nobel laureate in chemistry for his method to use carbon-14 for age determination. And this was a very scientific method, and it involved sampling of skeletal materials, skeletal collections at museums. At this time, there was very, very little discussion on the ethics of using radiocarbon dating and sampling. There were actually other Nobel laureates that has had a great impact on archaeology, as for instance, Frederick Soddy, who also was a Nobel laureate in chemistry for his contributions to our knowledge of radioactive substances and investigations into the origin and nature of isotopes. Also, then, of course, there's much less discussion on ethics and isotopes than it is on ethics and aDNA. we're getting closer now in terms of Nobel laureates, because Kari Mullis became a Nobel laureate in chemistry in 1993 for contributions to the development of methods within DNA-based chemistry and for his invention of the polymerase chain reaction method. Now we're really talking DNA, aDNA, and archaeology. Still very little discussion on ethics and DNA. And we're getting even closer now for this year's Nobel laureate in physiology and medicine, notably not in chemistry, but in physiology and medicine, where Svante Pääbo is going to be awarded in December the 10th for his discoveries concerning the genomes of extinct hominins and human evolution. And there's still very little discussion on ethics on human evolution in the long-term perspective. It's more a matter of ethics when we discuss more recent materials. So there has actually been several Nobel laureates with a close connection to archaeology, and we can almost regard this one as a prize in archaeology. So what is the problem then, ethics and archaeology? Why is there this divide between the humanities and the social and the natural sciences? Because there is always, or there has been for a long time, problems in the communication between the humanities and the natural sciences, I would say, in terms of aDNA. So in an article that I and my colleague Nina Eriksson wrote for the journal Current Swedish Archaeology a couple of years ago, we addressed this particular question. Why is it that natural science in the service, that's not maybe appropriate expression, of archaeology is so provocative or even threatening to some archaeologists? When I started working in archaeological science, I always had to fight to introduce natural scientific methods to archaeological materials. And now it seems so obvious that you can use these methods, but still it's not uncomplicated. Because up to this day, we can read upset comments on different blogs on, for instance, the latest findings on the application of aDNA to different prehistoric materials in the relation between the Minoans and the Methians, or whether the female warriors in the Viking, if there were actually female Viking warriors, etc. So we promoted the idea of a filter or a mental shield that comes between the scientists and the archaeologists, preventing fruitful communication. We find that the shield occurs both among the scientists and the archaeologists. It is thus not a one-sided problem. So our suggestion for a solution of how to remove this shield was to improve the communication between the disciplines by trying to speak a common language. And we did not argue that they all need to speak the same language, but a common language understandable to both the scientists and the archaeologists. And by referring to the filter, we were not interested in passing any accusations on any side. We were just trying to understand why this happens. So what happens then when you lack a common language? Well, I will just provide some examples with problematic opinions and uncomfortable terminology. In this department, my colleagues working on aDNA have been told that the humanities, people from the humanities, archaeologists, we are taking responsibility to our own knowledge production in a better way than you do. And then they use a lot of wordings that are maybe uncommon, uncomfortable for natural scientists regarding temporality, ontology, or and phenomenology. On the other side, there are these genetic researchers or the eDNA researchers that do talk about, have other terms that are uncommon or uncomfortable for archaeologists, as for instance, they do studies on polygenic risk scores. So what is a polygenic risk score? Well, it is defined by NIH as one way by which people can learn about the risk of developing a disease based on the total number of changes related to the disease. A person's polygenic risk score is a statistical calculation based on the presence or absence of multiple genomic variants without taking environmental or other factors into account. So I would like to show you a study that has been referred to previously in the previous presentations, and this is the study on the population genomics of the Viking world. And in this study, they do an analysis of the polygenic risk scores. And here they have compared the Viking Age sample against the present day Danish random sample. And what do they compare there? What do they look at? They look at age at menarche, age first sexual intercourse, hay fever, bone mineral density, body mass index, chronotype, hair color, black, adult height, hypertension, neuroticism, pulse rate, systolic blood pressure, schizophrenia, time spent watching TV, waist-hip ratio. Okay, what did they find? They didn't find any statistically significant difference for any of these traits, except for black hair. And since this is a method developed to look at different diseases, the expression is polygenic risk scores. In the text later on in this article, they say polygenic scores of Viking Age. And that they found statistical differences in 3 traits, but 2 they could take away 2 of the traits since they were not significant after accounting for the number of tests. What they found then that there was a difference in the, or rather they found that black hair color risk alleles were higher at the, in the Viking Age sample. So polygenic risk scores. Why do we even do this when we look at the different parameters they were looking at? Do we do it because we can, because we don't take the scientific questions seriously, or because we are ignorant to the scientific field? And now I would like to refer to Eske's introduction, that the questions that we ask to the material are extremely important. The questions are the reason why we're allowed to do this destructive sampling, etc. In my opinion, these analyses were performed because we can. I think it is problematic to look at age at first sexual intercourse as a genetic that you can find that in genes, because it's very, very culture dependent, I would say. I think that's actually bad science when you're looking at confounding variables in multiple correlations. But what is actually the scientific question here? I think that when you add on the parameter watching TV, I know it probably stands for something else, but it's phrased here as watching TV. It becomes ridiculous, and I don't want this kind of research to be ridiculous. I think it is extremely important. And I think it's an unethical phrasing to say that having dark hair is a risk. And I think also that we do this because we can. And also, as Eske referred to in the beginning, we have all this data that we can do something of. But is it necessarily so that we have to use all this data to pose or to analyze things that are ridiculous? There are quite often, we also saw a picture in Birgitte Skar's presentation of a reconstruction of faces. You can do that using DNA, and this is an example. You see here 2 faces. This is a reconstruction of an individual from the Pitted site Ajvide on Gotland, and this is a picture of me. And usually when people do these kind of pictures comparisons, they have a Greenland Inuit and they have Eske side by side, but I didn't want to put that up, so I put that myself instead. So this is the Pitted individuals, and the other individual is me. Actually, these 2, this analysis in this particular study that was sent for publication was removed by the reviewer, so it has not yet been properly published. So what I want to say with this maybe a provocative presentation or a discussion is that I really think it is important that we do these kind of analysis, and they develop constantly, and we never know where they will end up. In the beginning, it was very difficult. Actually, it was a discussion not on ethics, but it was a discussion in Swedish national radio on Svante Pääbo's initial publications on erroneous DNA analysis. But then they were explained that this is necessary in order to develop this field of research. But all in all, we want the research to be performed within an ethically sound framework that is accepted both within the natural sciences and the humanities. And we must think of the research question, and we also have a responsibility of how the data is used. And Martin, you brought up the Haak publication, and I think that I used that in my teaching, because it was actually used by right-wing religious groups in the US to prove that the nuclear family is the way we should live like. So, and I think that the sound framework has to be accepted by both sides. It's not enough to be accepted in one of these fields, and I think it's all our response. It is in the responsibility of all of us that see to that this will happen. So thank you for listening.
Thank you very much, Kerstin. That was great. And by the way, I think everybody, all the panelists have interpreted their roles perfectly. It couldn't have gone better in terms of your contributions, from Eske presenting us very technical aspects, Martin giving us the interpretive aspects, Birgitte giving us talking about issues of the relationship between the curators of material and the aDNA laboratories, and of course, you, Kerstin, framing the entire thing in the larger ethical concerns. So now we've run a little bit over what we had anticipated for this, but I think that's perfect, because I really appreciate. I think everybody did a good job in their presentations. We're going to have a little bit of an just open discussion right now before moving on to some questions from the attendees. By the way, attendees, if you have any questions, now is the time to start submitting them. We'll get to them in a few minutes. One of the points that I wanted to ask, and this is primarily for Eske and Birgitte, perhaps somewhat. As Birgitte mentioned in her presentation, the negotiations that went on between the Norwegian University Museums and the paleogeneticists in defining how data will be stored and how access would be made how it would be made accessible to people, and that seems like it was a very active process. I know, because I was a part of it. But I want to know from Eske, is that something that's common in your experience when you engage with cultural heritage institutions? Is there a very active engagement with how the data is going to be stored?
No, it's not. I think I have to say, I think that in Norway, to me at least, it appears that you are actually ahead of most other institutions in terms of engaging, and both in terms of, I would say, the legal aspect, the signing of agreements and expectations and so forth, right? So I would say, in my experience, it's not the norm, to be honest. It's the more common way. I think people just do it. In my experiences, there's like a memory of or memorandum of understanding, and it's not particularly detailed, and a lot of the things that has been brought up during these talks here are not really considered. So I think you are way ahead. On one side, of course, it can be annoying in the sense that it's time consuming, and everybody wants the study to happen, right, and take place. On the other hand, I think it's the right way forward. I have to say, I think it has to happen along those lines because, I mean, if you look at I think the ancient DNA history until very, very recently, and to some extent still happening, I mean, it is kind of I think almost anarchistic. I mean, it's like a bone slaughter or whatever you would call it, right? So I think it needs to be. It's very timely, these discussions, and I think definitely that the Norwegian approach could certainly be used as you can say as a kind of base for further development. Yeah.
All right, I've got two hands here, and you both came in at the same time, so Birgitte was to my left, so also Birgitte, what was your comment?
Yeah, I think we've come up with a good solution, Eske, when it comes to the last MOU that we made between the university museums and your institution. I think very many things are in place, but what we also see is perhaps a need for the university museums to think differently about the new time that we have. We have a law in Norway that's extremely strict and that tells about the Norwegian university museums that are managing something on behalf of the state. And we're taking that sometimes very literally in a way that this precludes us from storing results of DNA analysis or even other types of analysis in international databases. I think we have to adapt to a new world where open access is the norm. It is in EU, it actually is in Norway at the Research Council. And then we should rather think in terms of if they're particularly sensitive data, how can we protect these, as you mentioned, with the indigenous groups in these public or big repositories. I think what is the new challenge is more in relation to how we collaborate around the research that is going to be done. You know, you have these large big data projects with a consortium, with your research scopes, with your financial limits, whatnot, and your timeline. And we come in and we suggest that we collaborate very closely, as closely as we can. And we have certain issues that we'd like you to take a look at. The way I see it, this probably interferes with your big data project and your timeline and everything. We're going to try and find some money so we could actually do this research on the side, but it will take more facilitation and more meetings and more energy, for example, from yourself and your collaborators. So I'll be very curious to see how this works, if it does work. I think this is a fantastic situation, and we would get to know more about the involved Norwegian material or Scandinavian material, and also go in depth on certain sites and so forth. I don't know whether you have any thoughts about this, Eske.
Yeah, no, this is it's an important point, and I think just like with indigenous communities, really, it's the same trend, right? That you are trying to find common ground, and you're trying to, you can say, accommodate different interests in terms of what to look for. I think one thing which is really important that you on your side keeps in mind and understand is that the data analysis, right, of these data in most groups it's the, you can say, it's the bottleneck in terms of, you know, it's very, for many analysis, not all, but many number of analysis, it's very time consuming. You need highly specialized staff to do it. That staff is very difficult to get because a lot of them get much better offers from industry and moves into that space. And therefore, I think one thing that needs to somehow go into these kind of agreements is to make sure that, in my view, that also on let's say, if archaeologists, for example, or curators have specific questions that are very time consuming, that there's also in place some kind of funding for undertaking these type of analysis. So of course there's certain types that you can say all parties most likely agree upon and are interested in, right? And there's some budgets. Typically, you know, the money is coming from a grant, and that grant, you know, you have promised certain deliveries, right? And you can say the staff is allocated, is adjusted to those deliveries. So if you want to do additional deliveries, right, there needs to somehow be additional resources. So there's many ways upon one can, I guess, try to approach this problem. One is, of course, that you do the grants together already from the beginning, and you try to kind of make sure that the different aspects are covered. But there you have to keep in mind that you know, for most grants of the scale we're talking about to do this type of work, like the Viking study, it's enormously expensive. And we are talking millions and millions of kroners to undertake such a study. And the place where you often can go and get that type of money is you know, in medical related foundations. And you really can't, or very rarely can, raise that amount of money for within, you can say, a pure archaeological or anthropological framework. So so it means that you know, there is a, you can say, a challenge, if you want, to really get sufficient staff under those grants to deal with things like, let's say for a moment, kinship, for example, or something like that, right? There can be arguments for doing it. I'm not saying it never can be done, but it has, you know, in terms of your application, it's typically very medical oriented. And you kind of, you can say, the history part of it, if you want, is something you can argue for doing to the extent it informs you about something medical, right? Do you see what I'm saying? So it is a tricky part, and I think that one thing that really needs to be done is from all parties, or both from you can say curators, archaeologists, but also the same for geneticists, is an understanding of the amount of work that is going into the different parts of the process. I mean, I experienced it in my own group that people who are bioinformaticians, for example, pure bioinformaticians, have very little appreciation for the amount of time it takes to generate the data, and even less appreciation for the time it takes to actually dig out a fossil and interpret it. And the other way around, I often have experienced that archaeologists, you know, they see these type of studies as sending a sample for C14 data. And it's by no means like that, right? It's a way more complicated. It's not, many of these things are not, you know, just you put it in and then, you know, you get something out. And I mean, it takes development, it takes testing, it takes all kinds of things. So there needs to be a better dialogue there, and so there can be an understanding of what it actually takes in terms of time and resources to do the different parts of a project. Thank you.
Yeah, I think even for those of us that work with paleogeneticists, a lot of times our understanding of the process is very much informed by popular media more than it is by by actual firsthand knowledge. And so we're used to seeing CSI where or any TV show you can imagine, where they send off a sample for aDNA analysis and it comes back 5 minutes later as a book of the entire person's life. But anyway, Kerstin, you've had your hand up for a while now.
Yes, I would like to comment upon the because your initial question was regarding storing the amount of data and then making it open access. So I think we came a bit away from that topic. But I would like to say that in Sweden, I don't know how it's in Norway or Denmark or in Germany either for that instance, but in Sweden we have this that data handling. You have to have a data handling plan when you start a larger project, and all data should be treated fair data and should be open access, and we have large repositories that can be used, so that is not a big issue. I mean, you just have to do it, and then it should be accessible to anyone. We also have a facility called SciLife Lab, where you can where any archaeologist can actually approach them and ask them for help in performing DNA analysis, help with bioinformatician, etc., etc. So it's more of a democratic way in terms of it's not completely you're not depending completely on these big groups that do perform these kind of analysis. It's also a more democratic way. And I would also like to say that the funding system might also be a bit different in Sweden, because during the last 10 years, the projects that have achieved the largest funding in archaeology has been DNA projects. So the research group here at Stockholm University, where the Center for Palaeogenetics, for instance, they have, I don't know how many hundred millions of Swedish crowns that they have actually retrieved during the last years, and it has had nothing. It's within the applications. It's been archaeological applications, so it's not been in medicine or in natural sciences. So they have been extremely successful in doing this. So it's very much appreciated. This kind of research is very much appreciated in Swedish and by Swedish funding agencies.
Yeah, hearing you talk, that was going to be my first question: is how does that sort of thing get paid for? But
I can say that's definitely not the case in Denmark, or for that matter, the UK, where I'm here. It's there. It's medical oriented, if you want it.
I wanted to pivot a little bit towards Martin's topic, which I really enjoyed that discussion and or his presentation. And it's you know, one of the problems that has long affected archaeology is what are we seeing when we see the movement of material culture in space and time. And as Martin presented it, it's been variously interpreted as migrations of peoples and migrations of, you know, self-identifying groups of individuals. And to a certain extent, it seems like DNA has become just another layer in that old system. But I was wondering about this when it comes to these kind of narratives entering the public sphere. So we have a public who's not everyone, but if you look at the general average person's interaction with DNA, it's going to be through, as Eske mentioned, these commercial ancestry companies, and they traffic in the concept of genetically identifiable ethnicity, right? This kind of social identity as some sort of biological reality, and I'm wondering how the narratives that we create, which may be very nuanced, sort of reinforce that concept in the public mind of social identity as a biological reality, and not just ethnicity, but even race. I wonder if you have any thoughts upon that, Martin.
Yeah, I mean, as I try to make the point in my talk, I think this is exactly the kind of danger that always looms when you have this kind of perspective on things, which comes, of course, from biology and from biological relations between people, that you start to overemphasize that side of things, or underemphasize the way human actions and beliefs and values are somehow mingled up, of course, also with biological things. So it's a kind of, it pushes the attention, more towards the biological side, which of course is justified, but it can then lead to this kind of, well, especially when narratives are then put out who are simplifying things again, which of course they have to be done when you are engaging in public dissemination. You have to simplify. But I think, yeah, that's probably the main point. How are we able to convey a nuanced picture, still being engaging for the public and still being interesting for the public? I think that's probably the most crucial point. And I think that mostly we, as archaeologists in general, but probably also other disciplines, we need to be more skilled in this way. And maybe also, I think, what is a very good point that some colleagues have made recently is that I do think that there is a huge potential for nuance in the way we are telling our narratives, or we are conveying what we are doing. I think a lot of people want to know more complex stories about genetics, surely, but about archaeology too. So I think we need to be better to tell both complicated but also interesting stories. It's not a contradiction at all. I think, in the contrary, I think it should be a bonus that we're actually able to tell a quite nuanced, complicated story. But it's somehow something we need to be trained better in, or to train ourselves better in. To how do we actually bring this over to the interested public, which actually usually is very interested. Do not believe that people want, this kind of very simple stories.
Kerstin, yeah.
Yeah, I think it would be a mistake to think that people are unable to understand a more complicated story. I think it's a very good point that you make there. I think that, and also that we should be better at presenting our own story, and not always letting it into the hands of the journalists, but taking charge in that presenting our stories. I would just like to say a comment I forgot about when it comes to repositories. There's just a new project in Sweden that has been launched that is called, it's an infrastructure project. It's called Swedig Arc, in which all archaeological data that is produced by contract archaeology, by museums, by universities, old data that is now present in museums and at universities, will be collected in one huge database where you can retrieve any kind of information that you would need for your analysis. And I think that would also improve maybe the contextual information in all these DNA studies, etc., because it will be much more easily accessible. And one final comment, and that is about the Yamnaya culture. And I found it interesting with this reconstruction of the Yamnaya warrior, because the spelling that we're using right now is the Russian spelling of the Yamnaya culture. Maybe we should all change it into the Ukrainian spelling, where it's actually situated in Ukraine, and change the Y to a J instead. And then we can skip that warrior, this Russian warrior thing.
That's this aspect of communicating with the general public, obviously critical, and also our engagement with the media is critical. There was a book recently published called Ancient DNA: The Making of a Celebrity Science, which I can advise if you have any of you have the opportunity to read it. And it tracks the history of aDNA, and in particular its interaction with the media. And one of the interesting, I made a couple interesting points about the interaction with the media. One of these was the sort of the need for media-friendly results as a way of garnering attention. And to a certain extent, it was, at least it was depicted in that particular book, it was part of the way of keeping attention on the lab and keeping funding going and to the field generally. If you, what I call a quest for superlatives, the oldest example of this type of DNA, or the most complete type of that DNA. But then it leads to the issue that Kerstin was talking about in her talk, which is, are we doing these things because they're scientifically relevant, or are we doing them because we can? Does the field exist to answer scientifically relevant questions, or does the field exist simply for the sake of existing? And coming up with all these kind of what one might call flash in the pan results. And to a certain extent, obviously it seems necessary, and I think all scientists go through this phase where we have to, when we engage with the media, we have to come up with something interesting. But it also can give a very skewed view of what we do. And I was wondering, for example, Eske, your name has been on quite a number of large and well-known publications, and quite attention-grabbing publications. And how do you view your relationship with the media, the popular media, I should say, not the scientific media?
Yeah, I mean, I view it, the scientific process and the scientific paper, and you can say the media part of it, as two different things. I mean, the scientific paper is about purely about the science, right? It's about creating, you can say, as much as we can, believe we can get closer to what happened, right? To the truth, if you want, as much as we can and we're capable of, and so forth. And there's not really, you can say from my end at least, when we start a study, it's not about media attention whatsoever. It's about, you can say, the scientific achievement, if you want, and how we can, you can say, move in our understanding of a certain problem or a certain number of problems. When you then, you can say, get to the stage where the publication has gone through, and you start, you can say, the media part of it. I mean, you are facing, in my experience, a number of problems, right? I mean, or challenges, let me put it that way. I mean, the first part is you normally engage with a press officer from a university, and already their take on it is to simplify the story, right? So it already starts at that point. And if you take a study like the Viking paper that was discussed here, I mean, it's an enormous complex paper. There's many stories in that paper that we believe are of scientific relevance. Well, if you actually look at what did the media pick up, right? It boiled down to, well, there was fewer blonde Vikings than there's blonde Scandinavians today, right? And then there's a few maybe other more complex, they say a few more things, but you know, it's really, I mean, incredibly simplified. And yes, I agree, you know, that we have a responsibility, and I believe we are trying every time to give the complexity of the study. I'm also normally, when I deal with media, you know, I'm mentioning many of these big papers have multiple stories, right? And you're mentioning them, and but what happens is that you know, many media simply pick out one story out of 10 possibilities, right? They're only running with one, and so it's very hard to I think it's very hard to you can say direct in the sense. I mean, you can direct it in the sense in terms of your wordings, for example. I mean, an example was when we did the first Aborigine Australian genome, right? And we found this kind of early divergence of Aborigine ancestors compared to the ancestors of Asians and Europeans, right? There's many ways you can frame that, right? My way to frame it was, well, it's the survivors of the first human explorers outside Africa, right? That's kind of a positive spin, right? If you could have made another spin, it would be, well, it's, you know, diverse, it's more primitive, or whatever you would, you know. So it's also about your wording, and there you can I think do something. But it is, you know, it's the media thing is something you are not in control of. That's my experience. You're not really in control of it, and you know, so you can do your best. You can try to direct, but in the end of the day, it is that journalist from that journal or whatever, you know, that makes the choice, right?
I understand,
but I see it. I mean, I don't do papers for media attention. I mean, that's not the driver. You can say that the media attention I see, or media part of it, I see as a separate part, and it's actually in many times I think it's quite unpredictable what catch the media attention. I mean, we have had studies also in high-profile journals, Nature, Science, where there was very limited media attention compared to other studies in the same journals, where I think the scientific result was way more interesting, really, than some of the studies where it's then got a lot of media attention. So it's also, I think, pretty unpredictable, really, but I never think it should be the driver of what you're doing. I mean, first of all, it's unpredictable. And more importantly, you know, as I see it, at least, our primary goal here is to improve our understanding of the world, right? From a scientific standpoint. So, yeah.
Thank you. And I didn't mean to sound like I was accusing you of anything there.
No, I understand
that.
But it's just that of course, it captures. I mean, people are, you know, as somebody said, right, generally interested in the past, right? I mean, it's something. It's I normally compare it to, you know, arts, right? We all want it. Nobody wants to pay for it, but, you know. So it's kind of a similar thing, right? Everybody interested in reading about it, right? And this about the oldest and so forth, right? I mean, or the most complete or whatever. I mean, in some instances, I think it has a value, scientific value of saying it's the oldest. I mean, it's not like it doesn't have a value itself, right? I mean, if you're pushing the boundary for what is possible, right?
Yeah, I get it. I get that aspect of it. Yeah.
So,
thank you, Birgitte. I do have your hand up for.
Yes, I would just like to add to this conversation about public dissemination that I think the museums, at least, and not least the university museums, have an obligation to tell more than one story, if there's more than one story to be told. I think we have an obligation to stick to the scientific evidence that supports the narrative, and very often when it comes to DNA, there will be alternative explanations. And I think as we have a possibility of steering what we're presenting, I think it's very important that we actually do that, and do that to a much, much larger degree than we do today.
Thank you. Now we have one question from the attendees, and I'm going to read it out, and we'll see if it strikes a chord with any of you. It's not directed at anybody in particular. The attendee says, "I'm interested in hearing what the panel think could be the best way to challenge the misuse of research in general. For example, the way that Kerstin noted the Hack et al. Paper was used by right-wing groups to justify the nuclear family. What is the best way to tackle this kind of misuse?" Which is Eske. So Eske, for your first, I have two other hands.
It's me? Okay. Well, to be honest, my approach there, I mean, I have got death threats. I have got you know, from right-wing organizations. I got criticism from left-wing organizations you know, I both be, you know, I've been accused for both being a racist and a traitor of my own race, right?
Right. You can't win, you know.
So I can't be both, right? I mean, that's pretty clear. I would say, I mean, I think the only thing I can think of we can really do is what has been said here, right? I mean, it is, my take on it is, we are scientists, our job is to try to understand the world the best we can. And the results coming out, sometimes they are, you know, politically acceptable, but one group not politically acceptable by another group. And sometimes they can, you know, provocative into the political debate, etc. But I have to say, I see my role not as political, but by providing the kind of the guideline is, well, we want to get as close to the truth as we can. And then I think Birgitte was right in saying, well, what can you do then to kind of avoid you know, the misuse, if you want. I think there will always be misuse. I think we just have to accept that. I mean, whatever you find, you know, it can be used in different ways. And it will most likely be used in different ways. I mean, I've seen, for example, you know, there's even, you know, then a researcher sent me a quote, right? They had taken from, it was like a right-wing newspaper in the States, and then they just changed Native American with white European, right? I mean, so they actually took a real quote and then just changed the word, right? And then suddenly it looked like it was completely the opposite of what we found, right? Who were the first people in the Americas? I mean, there's nothing you can do about this. Mm-hmm. What you can do is you can go out and try as the best you can to tell the real story as you see it, and of course emphasize that story that you think are the important story to tell. And I, to be honest, I'm not sure you can do much else really, because I mean, there will be people who will misuse this. There will be people who will be upset. And I actually kind of changed my mind over the last few years that when I get a death threat or you know, somebody accuse me for being something, what I say to myself is, you know, well, you actually made a study had an impact. I mean, that's how I look at it, instead of just be one by in the huge pile of thousands and thousands of studies coming out. Well, it actually had an impact, right? Mm-hmm. Because some people are reacting to it, right? So, but that's just my take on it.
Yeah. That's a very good take. I've never received a death threat, so I don't know how I'd react to that. I don't imagine anybody would want to attack me for anything. Well, Martin, your hand was up, wasn't it?
Yeah, it was. I think the good thing about this question is that we basically have been talking about it already for quite a time, and so I would rather just re-emphasize what also Birgitte said. We have the responsibility to do dissemination and probably also take a little more control of it, in the sense that we have organs like museums, for example, who are, you know, very good places to present the complexity of things and, you know, different sides to things, different perspectives on the same subject. And in the same way, maybe also we should be better in also directly communicating to the public, not only over journalists, but directly. I know it's of course a time-consuming thing, but if you, for example, see the impact of you know, the Graeber-Wengrow book, Dawn of Everything, which is a book which basically is not so much definitely stating some stories, but rather asking a lot of questions and bringing a lot of discussions into different kind of audiences, which were not so much interested in archaeology before. I think this, I mean, not everybody is able to do it in that way, because you need a lot of communicative skills, but I think there are a lot of scientists who are able to do this, and maybe it should also be thought a little about how the research institutions are actually prioritizing the public dissemination, and maybe there could be more support, also time-wise, for at least some of the researchers who have the interest and talent, maybe also to communicate in a better way.
Excellent, thank you. Birgitte, did you have your hand up as well? You didn't, all right. Well, we're bang on 12:30 here, so I think we'll have Martin as being our final final respondent there. I want to thank everybody for attending. I'd like to thank Birgitte and Martin, Eske, and Kerstin for presenting and for answering the questions so thoroughly. I love this topic. I could talk about it all day long, so I had a really good time today. And I should also say one of those side points with regards to the very last question that we were dealing with and how to counter misuse. In our recently revised guidelines from the Human Remains Committee, we actually included a 1 point which we called unintended consequences, and that's where we challenge the researcher in the planning stage to think outside the box, think of what are possible abuses of your expected results, and not let that drive what questions you may or may not ask, but in the planning stage, come up with strategies for perhaps countering any of these abuses, so that when it happens, you're not caught struggling for an answer. But anyway, thank you everybody for your participation, and enjoy the rest of your day.